Herbicide, also commonly known as weed killers, are pesticides used to control unwanted plants. Selective herbicides control specific weed species, while leaving the desired crop relatively unharmed, while non-selective herbicides can be used to clear waste ground, industrial and construction sites, railways and railway embankments as they kill all plant material with which they come into contact. Apart from selective, non-selective, other important distinctions include persistence, means of uptake, and mechanism of action. Historically, products such as common salt and other metal salts were used as herbicides. However, these have gradually fallen out of favor, and in some countries a number of these are banned due to their persistence in soil, and toxicity and groundwater contamination concerns. Herbicides have also been used in warfare and conflict. Modern herbicides are often synthetic mimics of natural plant hormones which interfere with growth of the target plants. The term organic herbicide has come to mean herbicides intended for organic farming. These are often less efficient and more costly than synthetic herbicides and are based upon natural materials. Some plants also produce their own natural herbicides, such as the genus Juglans, or the tree of heaven. Such action of natural herbicides, and other related chemical interactions, is called allelopathy. Due to herbicide resistance, a major concern in agriculture, a number of products also combine herbicides with different means of action. In the U.S. in 2007, about 83% of all herbicide usage, determined by weight applied, was in agriculture. In 2007, world pesticide expenditures totaled about $39.4 billion. Herbicides were about 40% of those sales and constituted the biggest portion, followed by insecticides, fungicides, and other types. Smaller quantities are used in forestry, pasture systems, and management of areas set aside as wildlife habitat. History. Prior to the widespread use of chemical herbicides, cultural controls, such as altering soil pH, salinity, or fertility levels, were used to control weeds. Mechanical control was also used to control weeds. First herbicides. Although research into chemical herbicides began in the early 20th century, the first major breakthrough was the result of research conducted in both the UK and the US during the Second World War into the potential use of agents as biological weapons. The first modern herbicide, 2,4-D, was first discovered and synthesized by W. G. Templeman at Imperial Chemical Industries in 1940. He showed that growth substances applied appropriately would kill certain broad-leaved weeds in cereals without harming the crops. By 1941, his team succeeded in synthesizing the chemical. In the same year, Pokorny in the U.S. achieved this as well. Independently, a team under Judah Quastel, working at the Roth-Amsterdam Experimental Station, made the same discovery. Quastel was tasked by the Agricultural Research Council to discover methods for improving crop yield. By analyzing soil as a dynamic system, rather than an inert substance, he was able to apply techniques such as perfusion. Quastel was able to quantify the influence of various plant hormones, inhibitors and other chemicals on the activity of microorganisms in the soil and assess their direct impact on plant growth. While the full work of the unit remained secret, certain discoveries were developed for commercial use after the war, including the 2,4-D compound. When it was commercially released in 1946, it triggered a worldwide revolution in agricultural output and became the first successful selective herbicide. It allowed for greatly enhanced weed control in wheat, maize, rice, and similar cereal grass crops, because it killed dicots, but not most monocots. The locust of 2,4-D has led to continued usage today, and it remains one of the most commonly used herbicides in the world. Like other acid herbicides, current formulations use either an amine salt or one of many esters of the parent compound. These are easier to handle than the acid. Further discoveries: the triazine family of herbicides, which includes atrazine, were introduced in the 1950s. They have the current distinction of being the herbicide family of greatest concern regarding groundwater contamination. Atrazine does not break down readily after being applied to soils of above neutral pH. Under alkaline soil conditions, atrazine may be carried into the soil profile as far as the water table by soil water following rainfall, causing the aforementioned contamination. Atrazine is thus said to have carryover, a generally undesirable property for herbicides. Glyphosate was introduced in 1974 for non-selective weed control. Following the development of glyphosate-resistant crop plants, it is now used very extensively for selective weed control in growing crops. The pairing of the herbicide with the resistant seed contributed to the consolidation of the seed and chemistry industry in the late 1990s. Many modern chemical herbicides used in agriculture and gardening are specifically formulated to decompose within a short period after application. This is desirable, as it allows crops and plants to be planted afterwards, which could otherwise be affected by the herbicide. However, herbicides with low residual activity often do not provide season-long weed control and do not ensure that weed roots are killed beneath construction and paving. Therefore, there remains a role for weed killer with high levels of persistence in the soil. Terminology: herbicides are classified, grouped in various ways, e.g., according to the activity, timing of application, method of application, mechanism of action, chemical family. This gives rise to a considerable level of terminology related to herbicides and their use. Intended outcome: control is the destruction of unwanted weeds, or the damage of them to the point where they are no longer competitive with the crop. Suppression is incomplete control, still providing some economic benefit, such as reduced competition with the crop. Crop safety, for selective herbicides, is the relative absence of damage or stress to the crop. Most selective herbicides cause some visible stress to crop plants. Defoliant, similar to herbicides, but designed to remove foliage rather than kill the plant. Selectivity: selective herbicides. They control or suppress certain plants without affecting the growth of other plant species. Selectivity may be due to translocation, differential absorption, physical or physiological differences between plant species. 2,4-D, Mecoprop, dicamba control many broadleaf weeds but remains ineffective against turf grasses. Non-selective herbicides. These herbicides are not specific in acting against certain plant species and control all plant material with which they come into contact. They are used to clear industrial sites, waste ground, railways and railway embankments. Paraquat, glufosinate, glyphosate and non-selective herbicides. Timing of application: pre-plant. Pre-plant herbicides are non-selective herbicides applied to soil before planting. Some pre-plant herbicides may be mechanically incorporated into the soil. The objective for incorporation is to prevent dissipation through photodecomposition or volatility. The herbicides kill weeds as they grow through the herbicide-treated zone. Volatile herbicides have to be incorporated into the soil before planting the pasture. Agricultural crops grown in soil treated with a pre-plant herbicide include tomatoes, corn, soybeans and strawberries. Soil fumigants like metam, and azomite are in use as pre-plant herbicides. Pre-emergence. Pre-emergence herbicides are applied before the weed seedlings emerge through the soil surface. Herbicides do not prevent weeds from germinating, but they kill weeds as they grow through the herbicide-treated zone by affecting the cell division in the emerging seedling. Dithapir and pendimethalin are pre-emergence herbicides. Weeds that have already emerged before application or activation are not affected by pre-herbicides as their primary growing point escapes the treatment. Post-emergence. These herbicides are applied after weed seedlings have emerged through the soil surface. They can be foliar or root-absorbed, selective or non-selective, contact or systemic. Application of these herbicides is avoided during rain because the problem of being washed off to the soil makes it ineffective. 2,4-D is a selective, systemic, foliar-absorbed post-emergence herbicide. Method of application: soil applied. Herbicides applied to the soil are usually taken up by the root or shoot of the emerging seedlings and are used as pre-plant or pre-emergence treatment. Several factors influence the effectiveness of soil-applied herbicides. Weeds absorb herbicides by both passive and active mechanism. Herbicide adsorption to soil colloids or organic matter often reduces its amount available for weed absorption. Positioning of herbicide in correct layer of soil is very important, which can be achieved mechanically and by rainfall. Herbicides on the soil surface are subjected to several processes that reduce their availability. Volatility and photolysis are two common processes that reduce the availability of herbicides. Many soil-applied herbicides are absorbed through plant shoots while they are still underground, leading to their death or injury. EPTC and trifluoroalin are soil-applied herbicides. Foliar-applied. These are applied to portion of the plant above the ground and are absorbed by exposed tissues. These are generally post-emergence herbicides and can either be translocated throughout the plant or remain at specific site. External barriers of plants like cuticle, waxes, cell wall, etc. affect herbicide absorption and action. Glyphosate, 2,4-D and dicamba are foliar-applied herbicide. Persistence, residual activity. A herbicide is described as having low residual activity if it is neutralized within a short time of application. Typically this is due to rainfall, or by reactions in the soil. A herbicide described as having high residual activity will remain potent for a long term in the soil. For some compounds, the residual activity can leave the ground almost permanently barren. Mechanism of action. Herbicides are often classified according to their site of action, because as a general rule, herbicides within the same site of action class will produce similar symptoms on susceptible plants. Classification based on site of action of herbicide is comparatively better, as herbicide resistance management can be handled more properly and effectively. Classification by mechanism of action indicates the first enzyme, protein, or biochemical step affected in the plant following application. List of mechanisms found in modern herbicides. Epsps inhibitors. Compounds kill grasses. Acetyl coenzyme A carboxylase is part of the first step of lipid synthesis. Thus, Epsps inhibitors affect cell membrane production in the meristems of the grass plant. The acaces of grasses are sensitive to these herbicides, whereas the acaces of dicot plants are not. Epsps inhibitors. The acetolactate synthase enzyme is the first step in the synthesis of the branched-chain amino acids. These herbicides slowly starve affected plants of these amino acids, which eventually leads to inhibition of DNA synthesis. They affect grasses and dicots alike. The Epsps inhibitor family includes various sulfonylureas, imidazolinones, triazolapyrimidines, pyrimidinyl oxybenzoates, and sulfonylamino-carbonyl triazolinones. The Epsps biological pathway exists only in plants and not animals, thus making the Epsps inhibitors among the safest herbicides. Epsps inhibitors. The enol-pyruvyl shikimate 3-phosphate synthase enzyme Epsps is used in the synthesis of the amino acids tryptophan, phenylalanine and tyrosine. They affect grasses and dicots alike. Glyphosate is a systemic Epsps inhibitor inactivated by soil contact. Synthetic auxins inaugurated the era of organic herbicides. They were discovered in the 1940s after a long study of the plant growth regulator auxin. Synthetic auxins mimic this plant hormone. They have several points of action on the cell membrane, and are effective in the control of dicot plants. 2,4-D is a synthetic auxin herbicide. Photosystem II inhibitors reduce electron flow from water to NADPH2+ at the photochemical step in photosynthesis. They bind to the QB site on the D1 protein, and prevent quinone from binding to this site. Therefore, this group of compounds causes electrons to accumulate on chlorophyll molecules. As a consequence, oxidation reactions in excess of those normally tolerated by the cell occur, and the plant dies. The triazine herbicides are urea derivatives of photosystem II inhibitors. Photosystem I inhibitors steal electrons from the normal pathway through Fes to FDX to NADP, leading to direct discharge of electrons on oxygen. As a result, reactive oxygen species are produced and oxidation reactions in excess of those normally tolerated by the cell occur, leading to plant death. Bipyridinium herbicides inhibit the Phi-S, FDX step of that chain, while diphenyl ether herbicides inhibit the FDX, NADP step. HPPD inhibitors inhibit 4-hydroxyphenylpyruvate dioxygenase, which are involved in tyrosine breakdown. Tyrosine breakdown products are used by plants to make carotenoids, which protect chlorophyll in plants from being destroyed by sunlight. If this happens, the plants turn white due to complete loss of chlorophyll, and the plants die. Mesotrione and sulcotrione are herbicides in this class. A drug, nitisinone, was discovered in the course of developing this class of herbicides. Herbicide group. One of the most important methods for preventing, delaying, or managing resistance is to reduce the reliance on a single herbicide mode of action. To do this, farmers must know which herbicides share the same mode of action, but the relatively complex nature of plant biochemistry makes this difficult to determine. Attempts were made to simplify the understanding of herbicide mode of action by developing a classification system that grouped herbicides by mode of action. Eventually, the Herbicide Resistance Action Committee and the Weed Science Society of America developed a classification system. The WSSA and HRAC systems differ in the group designation. Groups in the WSSA and the HRAC systems are designated by numbers and letters, respectively. The goal for adding the group classification and mode of action to the herbicide product label is to provide a simple and practical approach to deliver the information to users. This information will make it easier to develop educational material that is consistent and effective. It should increase users' awareness of herbicide mode of action and provide more accurate recommendations for resistance management. Another goal is to make it easier for users to keep records on which herbicide mode of actions are being used on a particular field from year to year. Chemical family. Detailed investigations on chemical structure of the active ingredients of the registered herbicides showed that some moieties have the same mechanisms of action. According to Foruzish al. 2015, these moieties have been assigned to the names of chemical families, and active ingredients are then classified within the chemical families. Accordingly, knowing about herbicide chemical family grouping could serve as a short-term strategy for managing resistance to site of action. Use and application. Most herbicides are applied as water-based sprays using ground equipment. Ground equipment varies in design, but large areas can be sprayed using self-propelled sprayers equipped with long booms of 60 to 120 feet with spray nozzles spaced every 20 to 30 inches apart. Towed, handheld, and even horse-drawn sprayers are also used. On large areas, herbicides may also at times be applied aerially using helicopters or airplanes, or through irrigation systems. A further method of herbicide application developed around 2010 involves ridding the soil of its active weed seed bank rather than just killing the weed. This can successfully treat annual plants but not perennials. Researchers at the Agricultural Research Service found that the application of herbicides to fields late in the weed's growing season greatly reduces their seed production, and therefore fewer weeds will return the following season. Because most weeds are annuals, their seeds will only survive in soil for a year or two. So this method will be able to destroy such weeds after a few years of herbicide application. Weed wiping may also be used, where a wick wetted with herbicide is suspended from a boom and dragged or rolled across the tops of the taller weed plants. This allows treatment of taller grassland weeds by direct contact without affecting related but desirable shorter plants in the grassland sward beneath. The method has the benefit of avoiding spray drift. In Wales, a scheme offering free weed wiper hire was launched in 2015 in an effort to reduce the levels of MCPA in watercourses. Misuse and misapplication. Herbicide volatilization or spray drift may result in herbicide affecting neighboring fields or plants, particularly in windy conditions. Sometimes, the wrong field or plants may be sprayed due to error.